Okay, hello. I'm Hozumi Motohashi from Department of Medical Biochemistry. Today I'm going to introduce our research, which is going on in my lab. My lab aims to contribute to the realization of healthy longevity by understanding the biological responses to various environmental factors and the resulting pathologies when these responses fail from the perspective of redox reactions. We believe that as we age, that is, as our lifespan increases, the duration of exposure to environmental factors also becomes longer, and this often leads to changes in redox reactions within the body. Changes in redox status induced by environmental factors are fundamentally non-enzymatic reactions. In contrast, traditional biochemistry textbooks have devoted much of their content to enzymatic reactions, those regulated by proteins. To fully understand the mechanisms of response and adaptation to environmental factors, it is essential to consider both enzymatic and non-enzymatic perspectives. We believe that effects of metabolites and environmental chemicals on biomolecules, whether enzymatic or non-enzymatic, represent a key challenge in the post-genomic era. Now that genomes of many organisms, including humans, have been decoded, we consider metabolism and the modification of biomolecules to be central issues in post-genomic research. In particular, we believe that there remains a largely unexplored world regarding how metabolites and environmental chemicals affect biomolecules through enzymatic and non-enzymatic means. The stress response mechanism that I have focused on in my research, the KEAP1 NRF2 regulatory system, is a system in which non-enzymatic modification of the KEAP1 protein serves as a trigger for the cell to acquire antioxidant capacity. I have previously reported that activation of NRF2 exhibits anti-aging effects. For instance, in various experimental models, including Alzheimer's disease models, premature aging models, and physiological aging models, I have demonstrated that activation of NRF2 confers anti-aging effects. Now, let's go on to the introduction of our current research projects. The first project I would like to introduce today focuses on the discovery and elucidation of novel stress response mechanisms. We are currently identifying new factors required for NRF2 activation and analyzing their functions. In parallel, we are also investigating redox sensing mechanisms mediated by nuclear factors. Recently, we have begun to uncover a novel response mechanism to hypoxia. Well-known PHD-HIF pathway responds rapidly to hypoxia by stabilizing the HIF protein, which is a transcription factor. However, we have found that when hypoxia persists, a completely different mechanism becomes activated. So this heat map shows the time course changes in gene expression in macrophages stimulated with LPS. It reveals that the inflammatory response of macrophages differs between chronic and acute hypoxic exposure. Upon further investigation, we discovered that an enzyme involved in the vitamin B6 metabolism functions as an oxygen sensor, and that vitamin B6 dependent metabolism is altered under hypoxia. In chronic hypoxic conditions, a vitamin B6 metabolizing enzyme acts as an oxygen sensor, leading to the suppression of vitamin B6 dependent metabolism. And this newly identified hypoxia response pathway is currently under further detailed investigation. One of the metabolic pathways that depend on vitamin B6 is the biosynthesis of supersulfides. Supersulfides are characterized by having multiple sulfur atoms linked in sequence, which we call sulfur catenation, and include compounds such as cysteine persulfide and glutathione persulfide. The conjugation of glutathione with electrophilic toxins typically requires the enzymatic reaction enzyme glutathione S-transferase, GST. In contrast, glutathione persulfide, due to the additional sulfur atom, exhibits higher nucleophilicity and can conjugate with electrophilic toxins non-enzymatically. Interestingly, NRF2 not only upregulates the expression of glutathione S-transferases, GST, but also increases the levels of supersulfides. And this demonstrates using the fluorescent probe SSP4, which detects supersulfide species. Knockdown of NRF2 leads to a decrease in supersulfides, while knockdown of KEAP1, which means NRF2 is activated, results in the increase of supersulfides. Interestingly, supersulfide molecules are thought to be closely associated with organismal aging. As shown here in red, supersulfides decrease with aged mice. Furthermore, in CARS2 mutant mice, heterozygous mice, which exhibit reduced levels of supersulfides, an emphysema-like pathology develops with aging. The second project focuses on the elucidation of the physiological roles of supersulfide molecules. When glutathione trisulfide, which is a supersulfide donor, is added to cultured cells, we observe an increase in mitochondrial membrane potential, elevated oxygen consumption, and enhanced ATP production. Therefore, these findings highlight the importance of supersulfides in mitochondrial function. We are also generating and analyzing various genetically modified mouse models in which the production of supersulfides is suppressed. For example, when mitochondrial supersulfide production is specifically suppressed in neural tissues, we observe marked postnatal growth retardation accompanied by neuronal cell death. When mitochondrial supersulfide production is inducibly suppressed after birth, the mice develop severe anemia with a significant reduction in erythroblasts. The third project focuses on overcoming cancer, with a particular emphasis on identification of therapeutic targets for treatment-resistant cancers in which NRF2 is abnormally activated. We have previously demonstrated that aberrant activation of NRF2 alters the characteristics of cancer cells and contributes to their malignancy. Currently, our research is centered on understanding how abnormal NRF2 activation in cancer cells affects the tumor microenvironment. Notably, it has recently been reported that NRF2 activated cancers suppress anti-tumor immunity and exhibit resistance to immunotherapy. This project is a collaboration study with the Department of Thoracic Surgery, in which we analyzed tumor samples from lung cancer patients using single-cell RNA sequencing analysis. And here we compared the types of CD45 positive immune cells within tumors between NRF2 activated and non-activated cancers. The results show a reduction in the proportions of dendritic cells and CD8 T cell in NRF2 activated tumors, suggesting that indeed anti-tumor immunity is suppressed in NRF2 activated tumor tissues. So we are trying to understand how NRF2 in cancer cells regulate the anti-tumor immunity. All right, in summary, our goal is to elucidate the interplay between transcriptional and metabolic responses to environmental factors such as oxidative stress and hypoxia. Through these works, we aim to contribute to the control of cancer and chronic inflammation, and ultimately to the realization of healthy aging and healthy longevity. All right, so this is the last slide, and these are current members of my lab. I always try to make my members think by themselves and have their own words in the discussion. So I hope that students will cultivate the ability to think independently, express their thoughts in their own words, and approach information critically rather than accepting others' statements at face value. In today's society, overflowing with information across all professions, so this is an essential skill. To those from clinical backgrounds, I always emphasize the importance of becoming physician scientists and physicians who can see the molecules behind the diseases. Understanding the molecular mechanisms underlying health and disease leads to deeper insight and better care. And I also believe that research is a form of self-expression, similar to painting, writing, or music, composition. Research reflects how an individual interprets and gives shape to the fundamental laws of nature. While graduate school is a time to build fundamental knowledge, technical skills, and logical thinking, maybe with limited space for personal expression, but still, it is still important to value each student's unique ideas and perspectives. This is my way of thinking. And let me tell you another important thing. At Tohoku University, graduate students benefit from robust support systems. And also the Tohoku Medical Megabank Organization and the Advanced Research Center for Innovations in Next Generation Medicine work closely with the Graduate School of Medicine, offering an outstanding environment for research in human biology. So this is a very unique feature of our Graduate School of Medicine. All right, so this concludes my remarks. Thank you for listening, and I hope many of you will consider joining us at Tohoku University to create great science together.